Hey, good morning. How are you? Or not for you, good morning. For you, it's in the middle of the day. Can you hear me okay? Mm, if you cannot unmute, it could be for two reasons. Either maybe you're at the university Wi-Fi. Oh,
Here
there
we go.
you go.
Yeah, yeah. Sorry.
It's fine. How are you today?
Yeah, pretty good. Looking forward to the talk.
Yeah, it will be
Or to be
really.
given the talk.
No, it's really interesting. We're really looking forward to it. Um, How did your new year start? I hope everything was good so far in your new year.
Yeah, yeah, yeah. It's been good. I mean, it was quite nice to kind of get the story published right before.
Yeah.
Yeah. So right now, still kind of finishing up some other projects and yeah, busy, but good. Yeah.
Yeah, it must have been a more relaxing holiday season this year than probably the last few years.
Yeah, yeah, that's for sure. That's for sure.
Yeah, I can imagine. Did you finally take like vacation and went somewhere or?
No, actually, I did not. But
Oh,
no, we just stayed at home and had a good time there. So
well, that's good.
yeah.
Has
How
it
about yourself?
been raining a lot in the Netherlands or is it just?
Today we actually had some snow finally.
Oh,
It's yeah, I mean it's been pretty hot over here, but at least for the time of the year, which is not so good. But yeah, so actually I'm not unhappy with the snow, to be honest.
yeah, I imagine. My parents live in Germany.
Hmm.
Mm, I grew up there, so when I talk with them, they tell me. And sometimes, to be honest, I still watch news and from Germany because it's like 20 minutes condensed news and not like, I don't know, here news goes on forever and
Uh,
they just have a condensed good form of
yeah, yeah, yeah.
the
It's a bit easier
public.
to
Yeah.
keep track of what's going on, kind of.
Yeah, especially with Ukraine and so on. I feel like in the US it's not that much, it's still discussed, but I feel like it's more acutely discussed
Oh, yeah,
in the
yeah,
European news because it's the neighbor basically.
yeah, yeah, exactly. So yeah, it's kind of weird. It's like a 16-hour drive from here, basically. So it's like, yeah, it's pretty close.
Yeah, it's kind of scary, no? But I hope that Germans finally give their okay to provide the tanks, but we'll see.
Yeah, what I read is that it looks like it, right? So, well, let's see.
Yeah, well, let's see. um,
I'm quickly gonna grab some water if that's okay.
Yep. Yeah,
Yeah,
we have still 3 minutes, so you have
yeah,
time to
I'll
relax.
be right back.
Yep.
Yep.
Hey everyone. Thank you for coming. I'll I'm sharing right now everywhere that we are starting. I'll put up the slides in a minute, and in the chat I will put the paper that we are discussing today. And it will be a really interesting talk that I think has many applications to disease, everything related to neurons. And it's a really interesting new discovery. So stay tuned. And in the folder then that I'm about to share, you will find the the slides and then also two movies because for whatever reason it used to work to have like short videos in when you upload a PowerPoint presentation. But lately, for a while now, it doesn't work anymore. So we have to kind of upload the video separately, and Dr. Kole will then refer to when you, if you would like to see the videos, when to switch to which movie. So yeah, and we will start in around 2 minutes. Feel free to share the room if you think this is interesting for people, and yeah, I'm really looking forward to this talk and the discussion. Feel free to raise the hand also if you want to participate in the discussion. You're welcome to do so. Or if you can't, feel free to leave comments in the chat about the research and the topic here. And yeah, we will be starting in 1 minute.
Okay, I am back.
Great. Can you hear me okay? I had the red bar for whatever, the red bar means that the connection
Uh,
is not so good, but yeah, if
okay,
you
now I can hear you fine.
okay. Yeah, I should, like, my Wi-Fi should be fine, but okay, I think we can slowly start. People will still keep coming in, um, but I think we can start with the introduction and then go from there. So, welcome everyone to Science Society, and a special welcome to you, Cohen. And before we'll start, let me give the audience a short introduction so they get to know you a little bit. Dr. Cohen Cole, he's a postdoctoral researcher, and um, he went to university at the Saxion University of Applied Sciences, and then he did his bachelor degrees in Windesheim and his master in science at the Radboud University in Nijmegen. And probably I don't know Dutch, so I apologize
Yeah,
if
that's all
I'm
right.
like totally being totally horrible. Um, yeah, and he's a neuroscientist. He is very interested in cellular and molecular mechanisms that govern neuronal and also glial communication and plasticity. And yeah, that's basically, the overview of the topic we will be discussing today. And usually, before we start, we ask our speakers, how did you discover, I don't know, the passion for science, or that you thought you want to become a scientist? Was it something you always wanted to do since childhood, or did something you like spark your interest in science? Thank you.
uh, yeah, I never really had this like path or like idea like I wanted to be a scientist. And yeah, you mentioned I first started at Saxion, which was like laboratory school, which ironically I did not like. So I only did that for a year, and then I went to Windesheim for this to actually be trained as a biology teacher. So I've always had a good link with biology. I really love biology, but it was only after like more studies after that for my bachelor's and master's that I really got into contact with research, and that's during those lectures, yeah, that really kind of struck a chord with me, just to kind of disentangle what all these mechanisms are that make our bodies do what it does, and in particular, the brain is really interesting to me because yeah, that's in my mind at least, that's kind of really what makes us who we are and what we do. So yeah, that's So yeah, short answer is no. I never really had this plan, but yeah, during my studies, I kind of rolled into it. Let's say.
Nice. And you do electrophysiology mostly. I, to be honest, went to neuroscience. I always liked biology too, and I was deciding between a lot of things. But when I did electrophysiology in the lab, and I saw this, this little neurons in a dish like spiking
Mhm.
and doing stuff when we put different drugs on it. I don't know. I kind of really liked it, and I wanted to do that, like to see what neurons do when I do different things to them. It was
Yeah,
mostly the
that's
technique
also
for me, interestingly.
yeah, but it's also yeah, it's quite cool. I think as we'll also show, I hopefully will be able to show in my talks. Like if you measure these neurons, you can actually like immediately get responses, and you really have a sense that you're really working with these living cells that immediately respond to stimuli, and yeah, that's really cool. I mean, it's quite different from using like cell culture that you just I don't know throw some pharmacological stuff on, and then the next 2 days you see if they grow or not, or that's quite different. I I think.
Yeah, I agree. And behavior even worse than you're not supposed to look at it only when you do histology. So it sometimes takes months until you know if things work. So
Yeah,
that's
yeah, that's also histology is another craft. That's
yeah.
for sure.
Yeah, and then if you could bring us to then this project, how this project came about, was it really hard or to also get funding, or is there maybe an interesting background story regarding this project?
Um, Not really from my side, to be honest, because so my PI, Martin Kole, so we share, by the way, we share the last name, but just to be sure, we do not share family. We're not related. I get this question a lot, so I just wanted to kind of throw it out there. Um, but yeah, I mean, he already had a lab established, and he has already made a name for himself for studying kind of the role of myelin and also myelin loss in yeah, axonal signaling and neuronal excitability in the communication. And he was already kind of branching out to other cell types. So in this case, we'll be talking about the parvalbumin positive interneuron. So he had already had another postdoc working on that. But I'll show you also in the introduction why. But there was also a little bit of interest about the role of mitochondria in these cells. So it was kind of yeah, and for me in terms of like the techniques that he wanted to use and the project, it was kind of a perfect fit between what I wanted to learn and what I could already do, what I could contribute to his group. So that was nice, but like in terms of the funding, that's really was really his his doing, let's say. So it was, yeah, I guess kind of a natural expansion of his research line, let's say.
Yeah, we are very curious, to now to leading into the presentation to hear like the more detailed story about this project. And as I said before, for everyone that just arrived, um, there is a folder link pinned on top of the room, and there is a PDF file, and then also movie files. Um, yeah, as I said, now we have to separate them when we upload PowerPoints. Even small movies are not embedded anymore. Yeah, and Dr. Kuhn will refer to them accordingly. So the stage is yours. Thank you.
Thanks. Yeah, yeah. There's two short videos. I will mention them, but also on the slides, the relevant slides, there will be like a little note referencing them. All right, then let's get started. And okay, so I'm also new to this format, so I hope I will remember to mention to go to the new slide every time. But excuse me if I forget. So let's go to slide number one. So the introduction. Um, yeah, so this project is really focused on a particular type of interneuron, which is the parvalbumin positive interneuron, which is also known as a basket cell, which has to do with their morphology, which I'll come back to in a few seconds. And these cells are really fast spiking interneurons, so they are inhibitory interneurons. And then in figure one to the left, you see a trace of one of these cells, and you see that they can spike at really high action potential frequencies. And then to the right of that in blue and red. In blue, you see um, the dendrites and the soma of such a cell, and then in red, you see this massive axonal tree. So this is actually one reconstructed PV cell, and in red, you see this huge tree of the axon with a lot of different branches. And this, combined with the high spiking frequency, allows these cells to really effectively inhibit and control the surrounding neuronal network. And now what's interesting is that, as you can see in figure two in green, you see that these cells are myelinated. But the myelin is only found in the proximal regions of the axon. So the further away you go from the soma, the less myelin you find. And this actually means that a very large part of the axon is actually unmyelinated. And this makes it a bit curious, right? So do these cells even need the myelin if only such a small part of the axon is myelinated? But the answer to that question is yes, because in figure three at the bottom, there's a figure from a nice paper by Tsupi et al., where they actually looked at the number of interneurons in control patients or in multiple sclerosis patients, where actually myelin is broken down or is attacked by the immune system and then broken down. And what they found is that the PV cells actually are reduced in number in the MS patients, and they also see something similar for the somatostatin positive interneurons, which is a different type of interneuron, which sometimes are also myelinated, but not so often. But they don't see this at all in, for instance, calbindin or colretinin interneurons. So going back to the PV, parvalbumin positive interneurons, they do seem to be vulnerable to myelin loss. And also research in the Kole group from, I think, 2021 in eLife. There's also a paper from Mohit Dubey, where he also finds that these PV cells actually are inhibited in their or impaired in their ability to inhibit other cells after demyelination. Now, when I talk about myelin, what you probably think about is action potential propagation. That the myelin sheath is there to speed up action potential propagation and to make this whole process more energetically efficient. And that's absolutely true. This is a very important function of myelin. But what we also know, as you see in figure four, is that the myelin sheath actually also delivers nutrients, pyruvate and lactate, directly to the axon, which it is enwrapping, and these nutrients can then be used by mitochondria in the axon to produce ATP. So the myelin sheath has also a very important function in trophic support of the axon. And what's interesting is that these mitochondria are also known to be very sensitive to myelin loss. So, in what's typically observed is that in MS patients or experimental demyelination, what happens is that the mitochondria content of demyelinated axons starts to increase. And the idea here is that once your axon is when the myelin is lost, the action potential propagation is much more energetically costly. So you need more mitochondria to to help these action potentials propagate. So the idea usually is the more myelin you have, the less mitochondria you need, and the opposite. Um, but actually, we don't really know how this is the case in PV cells. So in slide 2, um, there's the research question that we have, which is actually how mitochondria are distributed and how they behave with respect to the myelin sheath, particularly in PV axons. Now, we were interested in this under normal conditions, but also after demyelination, so myelin loss. And to achieve demyelination, we use a toxin called cuprizone, which we can feed to mice for 5 to 6 weeks. And this effectively kills oligodendrocytes, and thus we can demyelinate the brain. And then on slide 3, on the left, figure 1, you see what that looks like. In red, we see PV cells. This is a PV-Cree TD tomato mouse. So all the Parvalbumin positive interneurons are labeled with TD tomato in red, and then in cyan you see myelin, and in control situation you see that there's quite a lot of it, and then after cuprizone it is very strongly reduced. If you continue the treatment, you can completely wipe it out even, but we don't go quite so far in this case. Okay, so that's how we basically can use cuprizone to experimentally test what happens after myelin loss. Then in figure 2, I show what we actually do to label the mitochondria specifically in PV cells. So I made a adeno-associated viral construct, so AAV construct, to express mitochondrial targeted GFP or mito-GFP. And if we then inject this into PV-Cree mice, or in their brains rather, then we can label the mitochondria specifically in these cells. So in figure 3, I show you what that looks like. As you can see, the expression of the mito-GFP overlaps very strongly with the TD tomato, already showing a very high cell type specificity. And what's nice is that we can actually make acute brain slices, meaning living brain slices. By the way, I'm not exactly sure what the background is of everyone, so I'm sometimes giving a little bit more information about what I mean. I hope I don't overdo it. If that is the case, just let me know. So if we make these acute living brain slices, what we can do is that we can also use patch pipettes to go into the slice and record from individual cells. And when we then stimulate these cells that express mito-GFP, we also see that they fire a very high frequency action potential. So this all goes to show that this method really allows us to label the mitochondria in a very cell type specific manner. Now, what's nice is that when we're doing these recordings, we can also fill the cell that we're recording from with myocytin. And then using immunostaining, we can actually kind of reveal the cytoarchitecture of this one particular cell. So on slide 4, I show you what that looks like. In figure 1, there's a Z stack of a reconstructed, well, not reconstructed yet, but a myocytin-filled PV cell. And then in combination with the mito-GFP expression in figure 2, I can actually map all the mitochondria within this cell. So as you can imagine, that's quite a lot of work, but it does give really nice data because we really then know what the shape is of these mitochondria and also where they are in the cell. So on slide 5, a little reminder of what I mentioned in the introduction. These PV cells are myelinated, but only at their proximal branch orders. So the proximal axon is myelinated. And when I say branch orders, what I mean is what I show at the bottom. The axon comes out of the cell, and then it branches into the second branch order, then it branches again into the third, then in the fourth and the fifth, and so forth. Um, so why is this relevant? Well, we actually want to know what happens to the mitochondria specifically in the proximal branch orders, which are typically myelinated. So in slide 6, then I can show you on the left an example of a control and cuprizone reconstructed axon. So this is an axonogram which shows the axonal branches, and every green dot is one mitochondrion. So again, a lot of work. um, then in figure 2, I quantified the density of the mitochondria in the entire axon. And between control and cuprizone, there's no significant difference. But then if we break it down by branch order in figure 3, what you see is that there's actually reduction in mitochondrial density, specifically in the proximal branches. And then in figure 4, there's also what we find is that there's a higher distance between individual mitochondria. So it seems like if myelin is lost, then so are the mitochondria. And on top of that, in the control situation, what you see in figure 3 is that the higher densities of mitochondria are found in the proximal branches. So this made us think that maybe these mitochondria are actually attracted to the myelin sheath in these cells. So to test this, we actually went back to the control situation, the control cells. So in slide 7, I show you a branch point of a control cell. And what's nice here is that these cells, because of their patchy myelin, their kind of intermittent myelin, what you can find are branch points where one branch is unmyelinated and the other is actually myelinated. And this actually allows you to have really an within cell control of the myelin, of the mitochondria content within the same axon. And if I do that, what I then find is that, first of all, these mitochondria are more elongated, so their aspect ratio is higher, and they're also larger. And within the myelin sheath, figure 4, then we also find that the density of mitochondria is higher. Now, this was really surprising because, as I said before, what is typically thought of, like the myelin sheath is typically thought of as the energy saver, right? So the more myelin you have, the less mitochondria you need. But in these cells, it actually seemed like the opposite was happening. But there was always something a bit pestering me, because what we're doing here is we're injecting a virus, which then labels the mitochondria, and then we're doing cell reconstruction and immunostaining. So there's a lot of steps in between that could introduce some kind of a bias. So I was actually looking for some way to have some independent confirmation of these results. So that actually brought me to a really nice website called Microns Explorer, microns-explorer.org. If anyone is interested, I can really recommend it. On slide 8, I show you what it looks like. So this is a data set of a square millimeter of visual cortex at the EM level, so electron microscopy data. So this is a huge data set. And what's really cool is that all the cells within this block, this 3D block, are annotated. And if you pick one of your cells that you're interested in and you double click on it, then on the right, you see that it's actually reconstructed in 3D. So because these PV cells have a very recognizable morphology, this actually allowed me to go into this data set, find PV cells, and then find axons that were myelinated or unmyelinated and then compare their mitochondrial content. So as you can see on the left, this black line, that is myelin. So that's very clear. And so these blobs within the axon, those are mitochondria. So both of these are very recognizable. So this is a very good way of yeah, testing this hypothesis that myelin actually attracts mitochondria. And on slide 9, I show a figure from a nice science paper from 2014, where actually they find that sometimes, well, not sometimes, quite often, layer 2-3 pyramidal neurons, so a completely different cell type, are also very patchily myelinated. So their axons have myelin, but very often, very large portions are actually not myelinated, or they have really short internodes. So we thought that maybe this clustering of mitochondria might not be specific for PV cells, but it has more to do with this patchy myelin pattern. So this also allows us within the same data set to compare PV cells and this other excitatory layer 2-3 pyramidal neurons with one another. So in slide 10, I show two examples of a reconstructed basket cell or PV cell on the left and a layer 2-3 pyramidal neuron on the right. And what you can, I think, already appreciate is that, again, there's more mitochondria under the myelin sheath compared to unmyelinated branches in PV cells. But this difference isn't so clear in the layer 2-3 pyramidal neuron. And also these mitochondria in PV cells seem larger. And of course, we quantified this in slide 11. I show you this in figure 1. Again, we find this higher density of mitochondria in myelinated branches. But we don't really find this, although it kind of goes in that direction, but it's not significantly different between in the layer 2-3 pyramidal neurons. And then in figure 2, we see that very interestingly, the mitochondria are actually much larger in PV cells, particularly the ones under the myelin sheath. They are much larger. And this actually also shows that they might be more efficient at producing ATP. Although right now I don't have any data to support it, but that would be my guess. And what we then thought was that, well, axons which are myelinated are usually a bit thicker than the ones that are unmyelinated. So it might be that if you have an unmyelinated axon, the cytosolic volume is less. Therefore, you need less mitochondria to produce enough ATP to fill up the cytosolic space with ATP. So when we corrected for that in figure 3, that turned out not to be the case. So again, in myelinated axons, the mitochondria took up much more cytosolic space in PV cells, but not in layer 2-3 cells, layer 2-3 pyramidal neurons. So this all shows that there's, first of all, a myelin-dependent clustering of mitochondria in parvalbumin-positive interneurons. But this isn't the case in layer 2-3 pyramidal neurons. So this also shows that mitochondria behave differently, not only within the same cell, but also between different cell types. Now, this was all really quite exciting and interesting to us. But as you may have noticed, everything I've shown you so far, everything is fixed tissue. So everything is standing still. But these mitochondria are actually also quite dynamic. And one of the functions that they have is that they buffer calcium. So when neurons fire action potentials, calcium starts to flow into the cytosol. And a few papers have already shown that mitochondria play a role in regulating the calcium levels in the cytosol by taking it up. They buffer it. But we had no idea what this calcium buffering, mitochondrial calcium buffering, looked like in PV cells, and also not with respect to myelin. So in slide 12, I show you how we tackled this. And so instead of the mito-GFP, what I'm expressing here is a mitochondrial targeted GCAMP, which is a genetically encoded calcium indicator. And what I then did is I went to one of these cells and filled it with both biocytin, just like I did before, but also with a red fluorescent dye, ATO594. And this revealed the cytoarchitecture within the living slice. And this actually enabled me to, first of all, firstly identify where the axon is, because as you can imagine, you see the dendrites and the axon, and everything is kind of moving. It's one big bundle of neurites basically. But what's nice is that their axons are really recognizable by very strong branching, and that they're also much thinner, and they have this very strong what do you call it? Uh, like a 90-degree angle branching, as you see in the bottom figures, and this you don't see in dendrites. So this filling of the cell with the dye also allowed me to kind of see where the axon is. And then because of the biocytin, after the experiments, I could find back the part of the axon that I was imaging, and then determine whether it was myelinated or not. So so what I did is that I drive these cells to spike and then record the mitochondrial calcium responses. And then in slide 13, I show an example of this. This also comes with video number 1. If the audience would like to see it, they can pull up this video now. What you see here is a part of the axon, and there's 4 mitochondria, and 2 of them are under the myelin sheath, and 2 of them are not. And then on slide number 14, you see what happens if we drive this cell to spike. You see that the mitochondria number 1 and 2, which are not under the myelin sheath, they show a very strong response to these action potentials. But mitochondria 3 and 4, which are actually in the same branch, but they are under the myelin sheath, they don't really show any response. So it seems that there is a myelin-dependent dampening of these mitochondrial calcium responses. And in slide 15, I quantified this between either in control cells, so either between unmyelinated axon or myelinated axon. We see that if there's a myelin, these mitochondria don't really respond anything, just like I showed you in the example. But then after demyelination, what happens is that the calcium responses start to increase slightly. Okay, so next what we wanted to know is whether this these calcium responses were kind of reflecting the cytosolic calcium responses. So basically to see whether this is really because of the calcium influx during action potentials. And in slide 16, I show you what we did. Instead of the GCAMP being targeted to mitochondria, we used GCAMP that is expressed in the cytosol. And then we basically did the same experiment. And then, as you can see in video 2, again, we see the same thing. So the calcium responses were really strong in, for instance, the AIS, which is where action potentials are initiated, and branch points, which are nodes of Ranvier. But in the internode, which is myelinated, there's actually no calcium response. And in slide 17, I actually show this the traces and also the population data. And as you can see, the cytosolic and the mitochondrial calcium responses are very similar. So actually, they show the same kind of picture where if there is no myelin, very strong calcium responses are seen. But if there is myelin, then these responses are dampened. And after myelin loss, there's again an increase in calcium responses. So this shows that the mitochondria are not only attracted to the myelin sheath, but their physiology is also regulated. Well, the activity-dependent physiology is regulated by the myelin sheath. Okay, and then on slide 18, I go through the conclusions. Yeah, so as I showed, in these PV axons, we see that the mitochondria are clustered to the myelin sheath and that they are larger, but we don't find this in layer 2-3 pyramidal neurons. So again, this is a cell type specific observation. Although I have to say that we don't know, for instance, what's going on in layer 5 pyramidal neurons, which are almost completely myelinated. We also don't know what's happening with somatostatin positive interneurons, which are also sometimes myelinated. So that's something also, although I don't mention it on this slide, something for the future to look at. So yeah, we actually don't know exactly to which extent this is a PV specific phenomenon. And yeah, we also showed that after myelin loss, this results in a mitochondrial loss in PV axons. And then on the dynamic front, or the physiological front, the mitochondrial calcium actually reflects the cytosolic calcium, and this is dampened by myelination. And after demyelination, we see an increase in calcium responses. Now, I already mentioned for the future, it would be good if we also investigate other cell types, but also I would be interested in ATP imaging in PV axons, because yeah, I mean, if there are more mitochondria under the myelin sheath, does that also mean that they produce more ATP? Or could it be that this is actually kind of a homeostatic mechanism or a compensatory mechanism, because there's less calcium flowing in, and calcium is actually one of the regulators of ATP production. So it might be that because there's little calcium flowing in, you just put more mitochondria there to produce sufficient ATP. We don't know at this point. Yeah, would also be really cool, I think, would be to do demyelination. And as I said, this takes 5 to 6 weeks. So you could actually follow in time what's happening to while the axons are being demyelinated. You could actually see what's happening to the mitochondrial content over time. And of course, yeah, we have no idea what the molecular mechanisms are right now that are targeting these mitochondria to the myelin sheath. So that would also be one of the questions that we'd like to answer somewhere in the future. Yeah, and that leads me to on slide 19 to thank a few people, in particular, yeah, the Kole group, Martin Kole, for really good supervision and great discussions and great mentoring, and then Bas, Maria, and Naomi for their help with all these experiments. I did a lot, but certainly couldn't have done it alone. So thanks to them as well, and thanks to the Verhagen group, who have been very accommodating in allowing me to use their molecular facilities to produce the viruses. And thanks to you for your attention, and I hope I'd be happy to answer any questions you might have.
Well, thank you so much, Kon. This was such a beautiful presentation and also really amazing work, and it must have been a lot of work. And congratulations for these findings, and then also to check if it's real a bunch of time in different ways. It's a really beautiful work. And when I read this, I was so puzzled that and then seeing now, your presentation and the videos, um, I was so puzzled that, calcium is less and they get attracted to the myelin. And do you have like an interpretation why that would be the case? Because I'm trying to think about that, like how the myelin, like does the myelin directly attract them? That's, discuss you want to check afterwards, or is it just because the calcium gradient and they just get attracted to that, the mitochondria? But if it's their job to basically regulate the calcium levels, why would they go to the place where there's less?
Yeah, 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 yeah. No,
So
that's
it's really
I
interesting
completely.
work because it's so many questions that it opens up. It's really interesting.
Yeah, I'm equally puzzled by this because yeah, like you say, mitochondria are known for their calcium buffering. So yeah, why the hell would you need them where there's no calcium? And um, calcium is actually also one of the things that is classically at least known to stall mitochondria. So for instance, you also find them very often at the presynapses, which also there's a lot of calcium flowing in there. So that was actually kind of surprising. But we also know that there's other cues that can stop mitochondria or kind of cluster them. One of them is glucose, and we actually don't really know whether glucose is directly delivered by the myelin sheath to the axon, although I think some of the like there's a preprint from a Klaus Armin Naves lab on bioRxiv right now where they do suggest something like that. Although, yeah, that still I think needs to be experimentally tested. And it could also be if glucose can stall mitochondria, then maybe pyruvate and lactate can also do something similar. Um, and it could also be that if there's, for instance, a high density of the sodium potassium ATPase, which usually like pumps sodium and potassium uh, in and out the cell to yeah re-establish the gradients. That's actually ATP dependent. So, and at least in white matter tracts in humans, it's been shown that that under the internode, so under sorry in the internode, so under the myelin sheath, you actually find a lot of this ATPase. So it could also be that this is actually a place where the ATPase is kind of clustered. So there's a lot of ATP production, ATP usage, and so you need more mitochondria there. Or yeah, like I said before, it could also be as simply that it's compensatory. That you do need some ATP in the internode, but there's not a lot of calcium flowing in, so these mitochondria are not stimulated a lot to produce ATP. So you just cram the internode with mitochondria. So even though they might not produce a lot, because you have many mitochondria, you will produce sufficient ATP. So I don't know. There's a lot of different angles to look at this. That all it's nice, but it's also difficult because it makes it difficult to kind of decide which yeah which one to tackle first and how.
Yeah, that is interesting with the ATP, that you need a lot of ATP maybe there. Another thing that came to my mind, but it's maybe far-fetched, that could it be maybe also the other way around, that the mitochondria kind of protect myelination for whatever reason, because it it could be there to buffer calcium in case, and it kind of protects, like it has kind of a preventive mechanism to not lose myelination because they are fast spiking, so it's really important for these cells, I guess, to not get intoxicated by too much calcium, and especially at those closer branches or more proximate branches. Um, yeah, I don't know. Do you think that could be possible?
Yeah, I can't say that I haven't kind of played with this idea as well. But I guess that would then, yeah, yeah. Another question kind of related to this is like that people have is like, what comes first? Is it like first you have the mitochondria and then the myelin comes, or is it you have myelination and that attracts the mitochondria? And I would say it's the latter, because after we remove myelin, then the mitochondria are also lost. So I suspect that it's the that it's the myelination that induces some kind of a attraction to the mitochondria. Whether it has a protective effect uh, for the myelination itself, I yeah, never say never, but I don't I wouldn't be so sure that that's the case, because yeah, that I guess also because the myelination is on the outside of the axon, so I guess then that would need some kind of a signaling then from the mitochondria to the to the myelin sheath, which is not impossible. But as far as I know, that hasn't been established. As far as I know, it has been established that there is signaling coming from the oligodendrocyte, so from the myelin sheath going into the axon. But I don't think it, at least it hasn't been shown yet, that it goes the other way around.
Yeah, yeah. I was thinking maybe they get some sort of reward type of signaling, like
Yeah, yeah.
both ways, kind of. But yeah, it's interesting that it seems for now to be different in the parvalbumin cells compared to other ones. But on the other hand, nobody has done such a thorough, you know, uh, so extensive experiments to analyze this interaction. So maybe we just don't know yet. Although you showed that it was shown that there's not such a big correlation between mitochondrial myelination and those type of pyramidal cells, but um, yeah, I don't know. Do you think it's a really distinct mechanism because of the fast spiking?
Um, I'm not sure if it would have to be the fast spike. Well, slightly. It might be related to it as well. But I have been thinking of maybe it's actually. I mean, again, like I don't know what's going on in somatostatin positive cells, right? So who knows if we look there, we might find the same thing, the same kind of pattern. I don't know. But I have been thinking of maybe it has also to do with parvalbumin, like the parvalbumin itself. Um, that is a calcium buffer that might also kind of influence how these mitochondria are situated. Um, yeah, it's a it's a it's a tough thing to really say much about right now.
Yeah,
Also
and
because yeah, because many things, most of the things that we do know about, for instance, mitochondrial motility and uh, how they are being anchored or distributed, those we often know from other cell types. So yeah, it's kind of difficult to say. And also yeah, like you said, some of the things that I found were already known, but then kind of distributed, yeah, like from different papers and different cell types, and yeah, so it would be good if for each cell type we have a really a good map of of mitochondria and the myelin sheath. And yeah, so much work to be done still.
Yeah, but this is the interesting type of work where you do
Yeah,
something and then it opens up so many more questions. And
yeah,
then
exactly.
in the chat, Aya posted a couple of questions. Have you looked at the role of matrix metalloproteases, MMP9, in demyelination? And then MMP9 is a biomarker in diseases such as MS, multiple sclerosis.
Um, no, honestly, I haven't. So yeah, that's also kind of what I said at the end. There's all kinds of molecular mechanisms that we'd have to look at to see what's exactly going on. Honestly, I'm not so familiar with MMP9 or the role in multiple sclerosis. I also don't know if it is say upregulated or downregulated. I don't know what usually happens in MS, but if this is also reflected in the cuprizone model. Um, but yeah, it might be a good lead. I will look into it and see if there's anything there that might be interesting for us to look at. But the short answer for now is no. I haven't looked at it, so I'm sorry.
Yeah, thank you. And I wanted to hand it over to Dr. Shah and ask, hey, Dr. Shah, do you have a question? Thank you.
Hi, Katerina. Thank you so much, Gwen, for your wonderful talk. And my question is about the clinical use of your research, as long as you just mentioned about the MS. And I was wondering, when we are talking about the MS, as you know, that we should think about different types, as well as we have a relapsing remitting and another type that is, which we call it the secondary progressive MS. So, and they have a different timeline, and I was just wondering, based upon your research, you think that if the drug want to develop out of your research, it might be beneficial in what stage mostly? And absolutely, the myelin sheath is gonna be, less and less by progression and everything. So, do you have any further information that you can share with us?
Um, so actually, I'm not familiar with whether PV cells in particular are, for instance, if they are more affected in one type of MS versus the other. So that I guess would be one thing to figure out first. I don't know if there's already data out there that I'm not familiar with. Um, but but I think if we can further understand um, how myelin and also myelin loss influences how energy use is affected by these yeah how myelin loss affects energy use in particular in PV cells. I think that would be very useful because these are very critical controllers of the neuronal circuit, and because of their high firing rate, they use a lot of energy. So yeah, that would be certainly something that I think would be useful also in a clinical sense. Um, I don't know if that answers your question sufficiently.
Yes, and because you just mentioned about the PV, and PV, as you just said, that in some of the researchers we have the immunoreactivity of the PV. And did you find that, I mean, in a case of the distribution in a very, for example, any kind of a specific part, that they are distributed more during your research?
Um, I'm not sure if I follow you. Do you mean the cells, whether the cell types, sorry,
Yes, I
the
mean,
PV
when
cells
you
are
want to think
distributed
about,
differently?
yes, integration, and you want to think, for example, we know that the PV, for example, they have an immunoreactivity in the frontal cortex, for example, in the frontal lobe. That's why we see the connection between the dementia, Alzheimer, and MS.
Hmm.
somehow, and that's why I was just wondering to ask you, did you
Yeah.
had any further?
Um, so yeah, to be honest, I mostly just focused on the somatosensory cortex, which is where we are doing our experiments. I am a bit hesitant to do any make any claims about any other regions. Although Mohit Dubi, my colleague, he has looked at the um, at least in the cuprizone model, right? I don't know about MS, but in the cuprizone model, he did not really see any reductions in PV numbers, at least in the somatosensory cortex. This could be different, of course, in for instance, prefrontal cortex. It could be that they're more vulnerable, and these cells start maybe dying or so. But um, at least in the model that we're using, we don't really see any changes in cell number, and I also didn't really see any axonal like morphological differences, which you actually do see in layer 5 pyramidal cells. So in the case of layer 5 pyramidal cells, which are very extensively myelinated, if you look at their axons after demyelination, then you really start to see, um, first of all, more collateral axons, like axonal branches being formed, but also they start to swell up. Like you see really big swellings of these axons, and those I haven't really seen in PV cells. Um, yeah, so no, I'm not really sure in terms of like their distribution, how that would influence the network. But as I alluded to in my introduction, we like previous work in our group did show that there is less that there's a loss of presynapses, so synapses from PV cells onto pyramidal cells. That goes down, so the overall amount of inhibition is reduced. That has been shown. Yeah.
And you mentioned about the lactate and pyruvate. And do you think that this contribution with the oligodendroglial is gonna kind of rescue the myelin sheath? Is that the role that you can just, I mean, consider for the amino acid that you just mentioned? What do you think about this part? Because there is some kind of drug that they are working with the pyrimidine synthetase inhibition.
Um, you mean whether lactate and pyruvate supplements could be
Yeah,
used to?
specifically these two because you just mentioned on your slide. I
Yeah,
was
yeah,
wondering.
um, yeah. That may well be. Um, Actually, that's one of the experiments that we were thinking of. One of the follow-up experiments to see whether if you locally apply uh, pyruvate or lactate to, let's say, an unmyelinated axon of a PV cell, if that is sufficient to cluster the mitochondria there. But at this point, we don't really we don't really know. Um, let's see. Yeah,
So
I
thank
mean,
you so much, Cohen, for your
yeah,
answers.
sure.
Yeah, I had a follow-up question. So when the PV cells are demyelinated, how long do they last? Did you like, and do they um basically age faster, die faster? Um, or didn't you wait basically to see how they do?
No, we always did our experiments after 5 to 6 weeks of cuprizone. Um, a previous PhD student in our group did extend that to 8 to 10 weeks, but he only looked at layer 5 pyramidal cells. So I at this point really don't know how that would affect the PV cells, but if the layer 5 pyramidal cells are any indication, then the cells don't start dying off or anything. Um, and also, when was doing the patch experiments, the recordings, then also it wasn't like these cells died much sooner or something, you know. So at least at this time point in the cuprizone treatment, these cells don't, yeah, at least on the physiological level, don't seem really unhealthy or something like that. But it might be that if you extend your the treatment long enough, then you might start to see really stuff happening to these cells because you get more and more activation also of microglia, and maybe more production also of reactive oxygen species and more damage to mitochondria. So all these things will, I think, at some point start to accumulate and accumulate. And yeah, I can well imagine that because these PV cells are so energetically costly, that they might be more vulnerable at some point. But at this point, I can't really say when that tipping point would be.
That's interesting. Yeah, that's really interesting because yeah, you would imagine that, I don't know, due to more calcium and maybe mitochondria working really hard, that maybe more NOS or something would at some point damage, but probably it takes a while. Like there's a good compensation going on, which is really interesting. Do maybe other cell types increase their activity to basically compensate for PV cells being kind of um, less myelinated? Did you,
Uh,
is there a way
well,
to, yeah.
yes. So in I'm not sure about SST cells, for instance, which usually inhibit PV cells, among others. But we do know from layer five pyramidal neurons, and this is actually kind of where this project, well, one of the projects started, because yeah, so previous work in our group after demyelination, we did see that there is an increased excitability of layer five pyramidal cells, and they really started to yeah, show kind of inappropriate action potential initiation. And yeah, from there on, the research line kind of grew to PV cells. And what I also, I didn't show it in this presentation, but I did show it in the paper. What we also show is that these PV cells fire lower amount of action potentials, so they're less excitable. So that is one thing that is which is different in terms of the connectivity, or like the, yeah, the network, let's say. But I'm not sure about any other cell types, whether they start also changing their excitability, or if there's any other um, homeostatic mechanisms going on. Yeah.
Yeah, it's, I mean, yeah, I would imagine that that they would fire less so, um, but yeah, it's interesting. Um, but so do you, so they are less excitable, but do you know like what the uh, why? Like, um, did you check which, you know,
Well,
I don't know which
we
receptors?
we did think maybe it has to do with some mitochondrial damage or something. Like there's less energy to have these cells, yeah, kind of reestablish their gradients or whatever. Um, but I'm not so sure if that's the case, because for instance, if I patch these cells, their membrane potential, their resting membrane potential, is kind of similar from control cells. So if there would be any impairments in ATP production, for instance, after cuprizone treatment, then what you would expect is that this would also start to affect the sodium potassium ATPase. So you would see, for instance, more depolarized cells, but that's not what we see. And also, if I look at the calcium responses in the AIS, so the axon initial segment, which is really where action potentials are being initiated. There, I don't see any differences in mitochondrial calcium buffering, for instance, and we also don't see any differences in the length or the location of the AIS. And both of these, so the length and the location, so the distance between the soma and the onset of the AIS, both can control the excitability of these cells. But yeah, it may be that what we haven't looked at is, for instance, ion channel composition. So it could be that there are, there's a difference in how sodium and potassium channels are being distributed in the AIS after demyelination. But I haven't checked that, so I'm not sure exactly what the cellular mechanism is that causes this lower excitability.
Yeah, that's really interesting that the resting potential is not different. So yeah, I would assume that it's voltage dependent. See going on, or maybe even something changes in the dendrites that whatever
Yeah, could
just
also be.
yeah
Yeah,
that
also yeah, another
they
thing that
become less
I
excited.
haven't looked at yet.
Yeah. Do you have calcium imaging, right? So you could do line scans in the dendrites maybe and check.
Yeah, I did a few of those, and yeah, there wasn't much happening actually. That was actually kind of interesting because there's also the mitochondria in the dendrite are much larger, and they're also I haven't quantified it, but they seem pretty densely populated in the dendrite. But then if you do the same experiments as I did in the axon, then there also some mitochondria do respond, but there's actually quite a lot that don't. So this also kind of goes back to what we talked about before, like these mitochondria under the myelin sheath, they may not respond to action potentials with calcium influx, but it doesn't mean that they're not doing anything there. So
Yeah, they're for sure doing something, and it will be really interesting to follow your work and see what happens because it's a really interesting puzzle to solve.
no, that's for sure. Yeah.
And then one last question from Einar that was posted in the chat. Can we see a different cytoplasma in the cell with the different mitochondria, and is electrical activity related to the cell organelle as a result of sugar intake?
I'm not sure what Einar means with the first sentence, a different cytoplasma in the cell with a different mitochondria.
Uh,
Um,
yeah, I think I don't know, Einar, do you want to come up and ask? But yeah, maybe okay. Let me
yeah,
invite.
but uh, oh,
Yeah, hi everyone. Um,
hey,
uh, um, this is not my work field, so be brutal. But can we see a different in the behavior? Is there a difference in the cytoplasma and the environment uh, when it comes to the cells with the mitochondria? As far as I get your presentation, that there are different sizes and they operate slightly different. Um, is there any cell organelles which actually behaves differently, or am I getting this wrong?
um, well, if I understand you correctly, um, I do know about the endoplasmic reticulum, for instance, which is another organelle, which very often has connections or contacts with mitochondria, and for instance, they exchange calcium as well. So ER is actually also a very important calcium buffering organelle, but besides mitochondria. Um, so that might be I can imagine that if you have larger mitochondria that for instance the chances of er contacts will also start to increase, and this might also influence, for instance, how maybe there's even local translation that is being affected in this way. I'm not entirely sure. um, then yeah then there's also microtubules which are necessary to um, which are necessary to transport mitochondria and other organelles to the place where they need to be. So I can imagine that in the myelinated axons, this might also look a little bit different. Actually, this has been shown. There's an e-neuron paper by what's her name? Um, Let me think. Oh yeah, Mitcheva et al. There they find that if you compare PV myelinated axons with non-PV myelinated axons, so different cell types, then also the microtubule density is different. Also, actually, they already kind of showed that there's a different mitochondrial protein expression there, but they didn't quantify the density and the volume and that stuff. And there were some other proteins that they found that were different in the PV axons that are myelinated compared to other myelinated axons. Yeah, so I'm not sure if this kind of uh, answers your question, but
it helps a lot. Thank you very much. Thank you.
great,
Yeah.
great, thanks. Oh, and
Yeah.
you also asked a question about the electrical activity.
Oh, yeah.
Do-do-do-do-do. Well, I mean, these mitochondria themselves don't really spike just like neurons do, but they do have a very strong membrane potential. And if there's a lot of sugar, glucose available, then this will drive ATP production, and this I think will also increase the membrane potential. So they have a very strong, like minus 200 millivolts uh, uh, membrane potential on the inside of their matrix. And this indeed would be influenced by the level of ATP production, which is which is driven by the electron transport chain, and that is actually what creates this negative membrane potential.
Thank you. Thank you.
Well, thank you so much for answering all these questions. And we've been going on for over an hour, so I wanted to give you an opportunity to go back to your life. and But thank you for sharing your really interesting work. I'm sorry for asking so many questions, but I was
No, no, no.
thinking
It's
so
it's my
much about
pleasure.
it.
Yeah, no,
And
it's really great. Thanks.
yeah, so I wish you all the best for the research and a lot of grants, because really curious to learn about, the next results.
Great. Yeah, thanks. I will do my best to obtain them.
Okay, good.
Yeah, thanks to everyone for their nice questions and the discussion and their attention. And hope to see you again.
Yeah, I hope we hear you all again one day. And yeah, and we are curious to follow your research, and maybe one day you come back and update us on, the future points you made that you are working on. And thank you everyone for coming. If you like discussions like this, follow the club. Our next discussion will be next week. Um, Dr. Ilinav will talk about phage therapy targets that that are only targeting harmful microbiome, like the harmful microbes in the microbiome, which I think will be really interesting too. So yeah, and thank you, Cohen, again. Enjoy your weekends, and yeah, thanks for coming. Thank you.
Yeah, thanks, and have a nice weekend, everyone.
Okay, I'll close the room in three,
Okay,
two, one. Bye, everyone. Thanks.
bye.